All right, I don't see anybody else in the waiting room at this point, so maybe we'll go ahead and get started. So I'll do a couple sort of housekeeping things first. So if you wouldn't mind, if you could put yourself on mute, except when you're going to ask a question or anything along those lines, it helps keep the background noise to a minimum. And if for some reason I start to freeze up, the easiest thing to help that is for you to temporarily turn off your video. So that you will still be able to see me and hear everything that's going on. But sometimes my bandwidth in Tacoma may not be enough to handle the number of people with all their videos showing. So for now, it's fine, I believe. But if that happens, I'll try to let you know about that so you don't lose me at some point. My name is Jim Gall, by the way. I'm at the University of Washington, Tacoma. I've met quite a few of you with my research crew as we've been out on the lake. And we'll all introduce ourselves here in a second. But just wanted to make sure everybody knows what they're doing. If you have any questions at any point, there's a couple ways you can do this. It looks like our group is small enough that we could actually just unmute ourselves and ask questions, so that's fine. Or if you would like, if you have done this before, there's a chat function. So down at the bottom of your screen, there should be a box that says chat. If you click on that, you can actually chat with us, with everybody, and ask your questions there. And I'll make sure that we pay attention to that as we go along. Right now, I've given you guys access to do things, and hopefully nobody Zoom bombs the meeting, and we'll be happy and can go on and actually talk to each other. That would be great. So that's the general scoop. Any questions on that end of things? All right. So what I will do to kick things off is I will share my screen here for a second, and then we'll do some introductions. All right. Thank you all for coming. So I wanted, since we, some of us know each other, but not everybody knows everybody, I thought it would be nice to introduce folks that are here. So first off, I was going to have folks in the UW Superfund research program and our research team introduce themselves. I will try to call off some names here, see if I can do this easily. Well, I'll try to go somewhat by memory. So Becca, you want to introduce yourself?
Hi, I'm Becca Newman. I'm co-leading the project with Jim, looking at arsenic in these lakes. And I'm a professor in Department of Civil and Environmental Engineering at the UW Seattle campus.
Thanks. Alex?
Hi, I'm Alex Horner-Devine. I'm also a professor in civil and environmental engineering, and I'm specifically focused on research about the hydrodynamics, so the movement of water in the lakes.
Erin.
Hi, I'm Erin, and I'm a research tech at UW Tacoma.
Excellent. Marco?
Hi, I'm Marco, and I'm a research tech at UW Tacoma as well.
Thanks. Sam?
Hi, I'm Sam. I'm a PhD student at UW Seattle, working with Becca and Alex to look at both the hydrodynamics and the biogeochemistry.
Great. Let's see. Is Tom here? I can't remember.
I think he's on the phone. Tom, are you here? Maybe not.
Okay. We'll come back. BJ?
Yeah, I'm BJ Cummings, and I am the Community Engagement Manager for the UW Superfund Research Program.
Great. Lisa?
Hi, I'm Lisa Hayward, and I manage research translation for the UW Superfund Research Program.
Excellent. Thank you. So that's, am I missing anybody else from our team? I think that's it. All right, we have some representatives from the Department of Ecology. If you want to introduce yourselves, Amy.
Hi, I'm Amy Hargrove. I work with the Tacoma Smelter Plume yard program. I'm the remediation manager.
Great. And Marion.
I'm Marion Abbott. I'm the acting section manager for the Toxics Cleanup Program in our Southwest Regional Office. And I'm also the project manager for the Tacoma Smelter Plume.
Great, thank you. I didn't see Justin. Is there anybody else from Fish and Wildlife here? Oh, I guess they couldn't make it. Kira for EPA Region 10, Kira Lynch.
Hi, this is Kira Lynch, and I am the Remedial Cleanup Program Branch Chief for EPA Region 10.
Great. And I think I saw Elizabeth from EPA.
Hi, I'm Elizabeth Allen. I work for Kara. I'm a regional toxicologist here at EPA Region 10.
Okay. I think that was everybody from EPA, if I didn't miss anybody. Public Health, Seattle, King County. Shirley?
Hi, I'm Shirley Tan. I'm the toxicologist for the Environmental Health Services Division in Public Health.
And Julie?
Hi, this is Julie West, Program Manager for that Healthy Community Planning Team for Environmental Health, Public Health, Seattle, King County.
Great, thank you. Then King County Environmental Programs, Chris Knutson.
Yes, hi, I'm Chris Knutson, work with King County Water and Land Resources. I run the Lake Stewardship Program at King County, and I'm the administrator for the Lake Geneva Management District.
Great. And Wafa?
Hi, everyone. I'm Wafa Tafish. I work with Chris in King County in the Science and Technical Support section, mainly on the Lake Stewardship Program.
Great. And City of Federal Way, Leah is here.
Hi there, my name is Leah Myrie. I'm the water quality coordinator with the City of Federal Way, and I also facilitate the lake management districts, including the Steel Lake Management District. I see John Pearson here. Thanks for joining us. There might be more on the phone. He's a committee member. And then I also see that John Hutton, our Parks Director, joined us, so I'll let him introduce himself to you.
Hi, everybody. Thanks for doing this. I'm John Hutton. I'm the Parks Director, and I appreciate the opportunity to participate.
Great. Nice to meet you. Thank you. I think I have all of our agency and UW partners. Did I miss anybody?
So Jim, I've been texting with Tom Burbacker. He's having trouble with his sound, but he had called in, so I'll just let folks know that he's with us as well. Tom is the director of the Community Engagement and Research Translation course, Lisa's and my divisions, and the deputy director of the Superfund program overall at UW.
Great, thank you very much. And maybe I'll do just a quick introduction since, and I didn't practice this one, but, and BJ, if you want to take over from me, but tell you just a little bit about the UW Superfund Research Program. So we're one of several centers around the US in different universities that work on research that's relevant to Superfund program or Superfund sites, Superfund projects. And so we have 4 different projects in our particular program. Ours is one of 2 environmental projects in there. Other ones, 2 are biomedical and 1 works up on the Duwamish, primarily on things related to fish up there. But we also have a whole bunch of different support cores that work with us on community engagement, research translation, analytical services, and other things, as well as training new students and people to do this work. So we are part of a bigger group, just so you know. So let's hear from you. So I'm going to try to rattle off people as I see them on my screen. So John, and if this is not your name, just tell us what your name actually is, but John Pearson, it looks like, is first.
Yeah, I'm on the Steel Lake Management District, and appreciate the opportunity to hear what you're doing. We've heard from you before, and so I'm anxious to hear the updates. And I see from time to time what's happening on the lake. I live on the lake, and so appreciate the opportunity to hear more and learn more.
Great, thank you. And I apologize, the street sweeper decided to go right outside my door for now. Norm, you're next.
Did you call me Jim? Oh yeah, I'm Norm Feast and I live on Lake Killarney and am involved in the Lake Killarney Improvement Association. And this picture you have here is right looking over towards my house. So that's a nice picture you have.
Yep. I see somebody just named Owner. I have no idea who that is.
That's me, Craig Rice.
Oh, hi, Craig.
I'm with the Lake Killarney Improvement Association. So I don't know why it says owner. I guess it's because my computer password's owner. So anyway, so thank you for doing this. I appreciate it.
No worries. Georgie's iPhone. Not sure. Let's see if we can unmute. Maybe not. So we'll come back to Georgie's iPhone if you can.
I understood it. Can you hear me now?
Yep, sure can.
Okay. I'm just a resident on Lake Killarney, just interested in what's going on.
Great, thank you.
Yes.
Mike Ficker.
There. I'm a resident on Lake Killarney and I got the giraffe in my yard.
Oh, we know that giraffe well. It's actually how we keep track of where we are on the lake. Sir Walken.
Yes, this is Dave Heimer with Washington State Fish and Wildlife. That's my that's my son's homage to Christopher Walken. So he got into my Google account.
Gotcha. I see Betty.
Yes, I'm not sure if you can hear me on someone else's computer, but this is Steve Ransom, resident on Lake Killarney.
Great, thank you. Anybody else that I missed?
Can we just ask Georgie's iPhone, is there a name that goes with that resident?
Georgie Heath.
Georgie Heath, great. Thanks, good to meet you.
Meet you. All right, wonderful. So so I think that's everybody's introductions. We would say that, and I'll point this out later, I think somebody, if you don't mind muting yourself again, I can hear some background noise. That'd be great, thank you. So just to let you know, Department of Health couldn't make it today because they've been redirected to support COVID-19 responses for the state. And so they have had input on this, but unfortunately couldn't be here today. And we'll talk about a little bit about what they've helped us out with so far. So we're going to give you a quick overview of the research that we've done at this point. You've seen some of it before, at least some of you have seen it before when we presented to Lake Killarney Improvement Association or the Steel Lake Management District, but some of this is newer and you haven't seen yet. So that's what we're going to spend time on. It'll be a brief overview. If you have any questions, please ask. And then at the end, after we present what we have, we'll have a time for question and answer, and then talk about some ideas for future work that we have and that you might have as well. So that's the scoop. So I'll get going here, if I can. Okay, so I'm going to give you an overview, and you've met several of the people that are on this list already who are working on this project or involved in the center. And our program is funded by the National Institute of Environmental Health Sciences Superfund Research Program. So what we'll talk about today is basically what makes shallow lakes like Lake Killarney and Steel Lake sort of special as far as arsenic goes, how that affects the arsenic that's in your sediment, how that comes out and potentially gets up into the water column where it can cause some issues with the biota. So we'll talk about the uptake into the biota and then the potential for human health risk that we've done some work on recently. So you all know that we've worked on Killarney and Steel Lake. That's why you're here. But this last go around, we added in basically what we considered to be sort of a background reference site. So that's Bonnie Lake out in Pierce County. And then we've been doing work on Angle Lake as a not shallow lake, as a deep lake that has high concentrations of arsenic. So we tend to compare the difference. What we're looking at is this difference between shallow lakes and deep lakes that may be contaminated with arsenic. And so Killarney and Angle have same concentrations of arsenic in their deep sediments, and we'll talk about that. Pine Lake is in here for anybody who's familiar. It's up in north part of King County. But Pine Lake we just use as a reference site for crayfish, so that we have kind of a background low-level arsenic lake that we can find crayfish in. So that's tougher to do. Bonnie Lake does not have crayfish. So this is all of that research in one slide. So I'll kind of step you through it slowly. So what we're looking at again is this difference between shallow lakes and deep lakes. And so the big difference, there are a couple different differences that cause the overall issues that we're interested in. First of all, shallow lakes get warmer than deep lakes do at the bottom. So shallow lakes, just because there's less water there, the light and the heat penetrates farther in. Anybody who goes swimming out in Lake Killarney or Steel Lake knows that it gets a little bit warmer towards the bottom. If you go out to Angle Lake and you dive down in Angle Lake, you're going to find that it gets cold at some point. And that's the point where you've reached the thermocline. So there's a cold layer of water at the bottom of Angle Lake that Steel and Killarney don't have as much of because they're shallower. Those warm temperatures that hit the bottom of those shallow lakes actually cause metabolism of the bacteria and things, the reactions that go on in the sediments of Lake Killarney and Steel Lake, they cause them to go faster. So bacteria works faster, and that results in higher amounts of arsenic getting released from the sediments through different reactions. Whereas in the bottom of Angle Lake, because it's colder, the reaction rate is slower. It's about 10 degrees Celsius difference between the bottom of Killarney and the bottom of Angle Lake. So that's one biggie. So temperatures first, and then the second one has to do with mixing. So these shallow lakes, they don't, and I know some of you are familiar with the word stratify or stratification, and some may not be. So stratification is basically that layering of the lake due to temperature differences. In this case, can be due to other things. But in this case, the temperature difference from top to bottom in Killarney doesn't develop as much. And so what happens is at certain times of the summer, when there's less wind and so on, you might actually get the lake to kind of set up short-term layering of the lake. You'll have just enough difference that the lake doesn't mix. And that allows arsenic to come out of the bottom when the oxygen's not present. Then you break down that on a regular basis in these shallow lakes. So either the wind comes up or just the day-night temperature differences causes mixing of the lake. There's lots of different potential reasons for this that Alex and Sam are working on. But basically you get this mixing of arsenic that was near the bottom of the lake, gets mixed up into the surface. In the deep lakes, like Angle Lake, you have that barrier, that thermocline actually prevents things. It sets up during the summer and it never goes away. And that prevents arsenic that comes out of the bottom of the lake from getting up into the surface much at all. So in those deeper lakes, you tend to keep the arsenic down below. And also down below, that tends to lose oxygen. And so you don't have many critters that are down there. You tend to have them up where there is oxygen. So that means in shallow lakes where you have arsenic coming up into the surface waters, where you have oxygen as well, that means now you have an overlap between arsenic in the water and your critters, and that results in the uptake of arsenic into those, or the enhanced uptake of arsenic into those organisms. Usually starting with phytoplankton at the bottom end of the food chain, or aquatic plants, and then moving up depending on who eats whom, right? So if you're an aquatic cow and you eat aquatic plants, or you eat plankton, then you're going to get more arsenic and so on. If that makes sense. On the other hand, in the deep lake, arsenic tends to stay at the bottom, and that prevents it from overlapping with the critters that are up above. So if any questions, one, you can either hold them or you can ask them now if you have any questions on that stuff. But that's the kind of basic science that results in higher arsenic in the surface waters in the shallow lake. It also results in higher arsenic in the shallow sediments in these lakes. So both Steele and Killarney has basically the same concentration of arsenic in their shallow sediments as they do in their deep sediments, because arsenic keeps moving up and then getting redeposited in your lake. And I'm about to get photobombed by my cat. I apologize. So, yep, science cat is on the prowl. So so overall, the new work that we did in 2019 throughout the year was we went out, and I know some of you probably saw us out there, we were collecting different organisms. So we had done phytoplankton and zooplankton before, which I think we presented before. But the new things that we looked at were snails, crayfish, and sunfish. So all of these organisms are things that humans can eat. Doesn't mean you have, and I'm guessing from the looks on some of your faces, not too many of you are eating snails out of the lake, but it is done. And so we're looking at, we tried to look at things that somebody might eat at some point. And what we find for all three critters is that as you increase the arsenic concentration in the oxygenated water at the surface of the lake, or you increase the arsenic in the shallow sediments near shore in those same lakes, the concentration in the organisms increases as well. So even though Angle Lake has the same amount of arsenic in the bottom of the lake that Lake Killarney does, it doesn't have anywhere near the amount of arsenic getting in the organisms because the arsenic's not getting up into those surface areas. So hopefully that makes sense. So the same thing. So these snails are kind of, you may see them out there, they're golf ball size. They're called Chinese mystery snails. They're actually an invasive species that's found kind of throughout Washington at this point. But people do harvest those to eat them. They also farm them to eat them. Crayfish, so there's not as many crayfish in Lake Killarney as some of you who volunteered to help us find them know. We put out a lot of traps, and there are crayfish there, just not as many, but there are some crayfish there, and we know that Steele Lake actually has quite a lot of crayfish in it in certain parts, and so there was a mix of those there. And then the sunfish in this case are either bluegill or pumpkin seed sunfish. So it depends on your particular lake. I believe that we found primarily bluegill in Lake Killarney, and we found pumpkin seed sunfish in Steele Lake. In the other lakes, it was either pumpkin seed sunfish or a mix of both. So this is the big stuff that we found this year, was that the concentration goes up as in these organisms as well. Well, then you might ask yourself, well, I'm sorry, this is just a summary to show that. So if you look through all those different organisms that we have data for in both Angle and Killarney, you can see that the concentration in Lake Killarney is higher than the concentration in Angle Lake for every organism. And this is on a log scale. So these are roughly 10-time difference between one lake versus the other. And again, that has to do with the difference in how arsenic gets out of the sediment and up into the water column in the shallow areas. Okay. So what does that mean for potential risk? What does it mean to be higher or not? So what we did was taking the concentrations of arsenic that we find in those different organisms and using ingestion rates or consumption rates that we have from EPA, I believe it is, that are different amounts of eating an organism. So they have ideas for how much the general population, so the mean consumer would be the average population in the adult population in the US, how much do they eat, right? So that's what the difference is. all of the organisms for each lake are based off the same amount of arsenic. It just depends on how many of that, how much of that organism you eat, right? So the more that you eat of that, the more potential risk there is from exposure to the arsenic that's in that. So you can see that the colors tend to go up as you go from mean consumer, which is like the average of everybody, which also includes people that don't eat any snails or fish, then into the 90th percentile, which is usually considered to be protective or considering what sport anglers or people that actually eat more of those would be in the population. And then the 99th percentile is for people that tend to eat much more. So subsistence fishers, people that are eating these things on a regular basis. So that's kind of the breakdown of what those mean. Usually the 90th percentile is where most of the risk assessment work kind of bases it on, but it can change depending on the particular situation being considered when people are looking at these, when the Department of Health is looking at these. So what we find is that the color coding, by the way, the orangish yellow color, so this is for cancer risk, and the orangish yellow color is for increased cancer risk over 1 in 100,000, basically. So 1 in 100,000 increased cancer risk for that sort of risk level, which is sort of a baseline risk level that people use most of the time, not all the time. The oranger color, I don't know, these are pastels, so it's hard to tell what exactly I'm looking at, but the middle color, whatever that is, that's 1 in 10,000, so the risk goes up by 10 times. The red color, that's 1 in 1,000, so the risk goes up even another 10 times. So the higher that color, the more the risk. So what you can see is if we look at the 90th percentile as one benchmark for people who are out there, sport anglers and so on, that are occasionally catching crayfish or fish or snails, you can see that snails actually have a pretty high risk if you're consuming snails. And that's because the snails are the lowest down on the food chain. So the snails are eating plankton and periphyton directly, and so they're getting quite a bit of arsenic that's getting into their tissues. And with snails, you tend to eat the whole snail. If you've ever had escargot, you more or less cook the whole thing and then take it out of the shell and eat it. So this risk level is highest for the snails, and you'll notice that even our reference site ends up with high enough concentrations of arsenic in Bonnie Lake to throw up a flag. So snails are taking in even background levels of arsenic at kind of elevated amounts, okay? The highest risk that we find for any of these 4 lakes is in Lake Killarney, because again, it has highest concentrations in the sediments and because of its shallow nature, as we talked about before. For crayfish, you can see that Steele and Killarney both have greater than 1 in 100,000 cancer risk, increased cancer risk for crayfish. And again, Killarney doesn't have many crayfish, so the potential danger of eating too much of that is less, but we do know in Steele Lake that there are people that go into Steele Lake specifically to catch crayfish. It's even on, there's a crayfish fishing group that's on Facebook, and you can see Steele Lake pop up occasionally for a place to go. Then for fish, which is the more likely thing that people might be going after, you'll notice that none of the lakes are actually above at the 90th percentile, but for populations that are potentially eating more fish, that 99th percentile, Killarney does actually have a higher risk than that 1 in 100,000. All the other lakes are below. And the reason again is that as you go, let me go up for a second, as you go up in the food chain, the amount of arsenic and the amount of inorganic arsenic, which is the more toxic form, actually decreases as you go up the food chain. So things that are farther down on the food chain have higher risk levels, things that are farther up have less. And then it depends on how much you actually have in your lake bottom as well, right? So how much is there to feed everything else. So that's the gist of the risk calculations we have. Now, Department of Health, Dave McBride at Department of Health kind of turned that into a, so what does that mean scenario for how much you would eat, right? So it's hard to kind of imagine what this means. So what he calculated was, and he said it was fine to share this, that for snails and crayfish, that if you had one meal a month for snails and crayfish from all of these lakes, that you would actually have more than the 1 in 100,000 cancer risk. So that's because the concentrations in those are higher. So basically, if you're eating, one meal a month or more, then you're at higher risk because of that. Makes sense? Then for sunfish, because the risk level was already lower, we know he calculated that 2 meals a month from the sunfish in Lake Killarney would result in a greater than 1 in 100,000 cancer risk. So that's where it gets more, I think, applicable to a lot of people who are out there catching fish, maybe taking them home. I know that we're out on both of these lakes. We see anglers out there on a regular basis, either in their boats or from shore. So we know people are catching these fish and taking them home, but we don't know how many times they're doing that or anything along those lines. So that's the basic health risk calculations. Now, Department of Health is considering next steps with this information, but they've gotten, unfortunately, as I mentioned before, they've gotten pulled away to support all the COVID-19 response that's going on. And they'll take our data into account, but they need to look at other things as well, and then they'll determine what they want to do next. So that's kind of a stay tuned as they get a chance to actually breathe and come back to this. They will be figuring out what to do next. What we're looking at potentially for next steps ourselves from the research group. So we've talked to both Department of Health and Department of Ecology, and if they have, as they look at additional information and potentially collect more data, we'll be assisting them with anything that they need as best that we can, but they will be kind of looking at this for any potential future things that they want to do, and we'll just be assisting them with try to hand off information as best that we can. We've talked to Department of Fish and Wildlife in a meeting recently, and there was a question about, well, what about other fish species that are in the lake? So we are going to be looking at some of the other fish species that people might catch in the lake to see if there's any difference in the amount in the other organisms like bullhead, or we expect that things like the stocked rainbow trout would actually be lower in concentration just because they're not living in the lake for very long. They tend to get stocked in and then get fished out relatively quickly. But other species might have higher or lower concentrations, and we want to look at that. And then future work that we're proposing at this point to do next is, we recognize that we don't want to come in and tell you, hey, you've got arsenic in the bottom of your lake, and then walk away. So one of the things that we know is that dealing with arsenic in lake bottoms is not easy to do from a cleanup standpoint. And so we want to begin some research on trying to figure out different ways to do that might actually be affordable and effective at the same time, but that would just be getting going. We want to try to come up with the tool that helps lake managers, public agencies estimate what the potential risk is for other lakes besides the ones that we've been studying. The great thing about being able to work at Steele and Killarney is it's given a lot of information that allows us then to sort of give that out to other folks and figure out what's the potential dangers in your lake or not. And then, so we'll create a tool that helps people to do that. And then there's also a question, as many people have asked at public meetings in the past, well, what about the organisms in the lake themselves, right? Does this have some impact on the birds or the fish or anything along those lines? And so we're trying to come up with some indicators or a better knowledge of what impacts the arsenic that we see get into the organisms might have on the organisms themselves. So that ecological health idea. So that's what we have. And so it's time for questions. And I think it's a small enough group so you can, if when you have questions, you can just unmute yourself. But if you don't feel comfortable asking that, you can email me a question either now or later. And I know some folks might not have been able to make it. So we will make this meeting recording, which it's doing right now. We'll make that available and send out the link to you folks so you can share it with other people that might be interested. And people can email me with questions and I'll feed them to the appropriate people if I'm not the person to answer it. So I'm going to stop sharing my screen right now, and so we can see each other a little bit better, and open it up to any questions that anybody has.
Hi, I apologize I was late, but was the impact on swimmers discussed? And thank you.
Thank you. The impact to swimmers was not discussed. I would say that at this point, we don't have any data on impact to swimmers. And so I don't know if anybody from any of the other agencies wants to field that question, but I think we can provide what data we have, and then it would have to be looked at in potentially with some additional data to try to make any sort of health risk determination for swimmers. Anybody else want to chime in on that from the agencies? Okay. So that's, sorry, Mark, that's a, we'll have to figure that out and get back to you idea. Other questions?
Well, in terms of, if I identify arsenic in a lake, what, if anything, are the remedies, or is there an ability to remove it? Is that recommended? Is that advisable even? And at what point would you recommend trying to remove it? But then how feasible is it to remove? I guess there's several questions there I'm asking, but.
I can answer some of that, but I think this might be something where maybe Marion or wants to step in and answer from ecology's perspective.
Can you repeat the question?
Well, yeah, if there is identifiable arsenic in a lake, for example, Steel Lake, that you've been studying, when is it advisable to consider removing it? And then what are the methods for removing it? And is it even feasible or advisable to do that?
Well, from a cleanup perspective for sediments, at least the technologies we tend to look at are pretty straightforward, that you either would dredge it out or you might cover it over with clean sediments and bury it, cap it down. I haven't looked closely at this information to know what kind of options might be the best. And I know that Jim and his group are looking at, are there some other techniques, that aren't, as physical, like removing the sediments or burying them over. And so I'm interested in what the university can come up with.
Great. Yeah, and just to follow up on that, that's what we recognize, because I do quite a bit of work on things like phosphorus removal or treatment in lakes, and arsenic and phosphorus kind of act similarly, or they are very similar chemically. And so it turns out that, to deal with phosphorus in the lake sediments, dredging is sort of one of those options to actually get rid of it. There are options for decreasing it coming out of the sediments, which would actually improve the situation, as you might all be aware of. A lot of those remedies don't work for arsenic because of the loss of oxygen at the bottom. And so what we're hoping to do is to develop something that we can put out on the bottom of the lake that would basically capture arsenic as it tries to leave the sediment. That's our proposed idea. Now, are we going to be successful? We'll find out. We have a good person named Professor Jessica Ray, who's done similar types of work in developing these before. And so we'll see how it goes, but that's going to be several years down the line as we help develop this stuff. Becca, do you have something you want to insert there?
Yeah, I just wanted to emphasize that we are in the very early stages of conceptualizing this. And so it is just a very kind of prototype idea right now. So I think it is, as Jim says, multiple years, many years away from any kind of actual implementation.
Yeah. Thank you.
Thank you.
Norm, you had a question?
Yeah, I had a question. When you did the study here, it seemed like most of your equipment was down on the south end of our lake, and it's sort of like two lakes in one.
Mm-hmm.
Did you do any study up at the north end?
We've done some in the past, Norm, but we didn't focus on the north basin of the lake, just because of the difficulties, as you know, with all of the aquatic weeds and kind of dealing with those. We definitely haven't done a thorough look at just how that acts. We expect that it's similar, but we don't know for sure. I do recall in the past that I actually collected at least some sediment samples from the north basin of the lake, and the concentrations are similar to what we find in the south basin. But we haven't done any of, as you saw, like all of our equipment's in the south basin just because of access issues and other things. So, but it's a good question. We are assuming it acts similarly, but it is actually even shallower, as you know, than the rest of the basin. So it may or may not stratify as much as the south basin does. So it has to stop mixing enough that the oxygen can get used up at the bottom of the lake, and that releases the arsenic. So if that's not happening in the north basin, then there's less arsenic actually making it into the water column possibly.
Yeah, a lot more weeds up there too.
Yep, yeah, quite thick.
Hi, Jim. One more. A couple of years ago, when you spoke to our group, you mentioned that the stuff tends to congregate in the center of the lake and not so. I'm worried about stirring it up. So is that still your assessment that most of it has moved to the center of the lake, and you have to worry about stirring it up around the shore if I am swimming, that kind of stuff?
Gotcha. It turns out it's a good question, but that's, as I mentioned somewhere, but definitely could have gotten lost, is that we are finding that the arsenic gets released from the bottom of the lake up into the surface waters, but then it actually gets spread out horizontally and redeposited. So we're finding the same concentrations in the shallow sediments around Lake Killarney and Steel Lake as we do in the deep sediments, which is very different than what we see in the deep lake, Angle Lake. So we actually are finding 200 parts per million of arsenic in the shallow sediments this is about 3 feet deep or so is usually where we sample. So it's not the immediate near shore, right, where people are, dipping their toes in, but it is pretty near shore. At this point, we don't know as much about the potential for stirring that up or the sort of what that would do as far as concentrations in the kind of, if you're wading out into the water, we don't know what that would do at this point. So it's definitely something that I think would be interesting or would be useful for us to look into. But right now, unfortunately, I can't answer that.
I have another question.
Oh yeah, go Norm.
Couple people on this lake have used some of that stuff. I don't know what we call it, muck eater. Something you throw in there and it starts digesting. I don't know exactly what it is, but it's something. Would something like that be useful? Because I've used it off my beach and it does seem to work and eliminate some of the muck. And I just wondered if that might be an answer to at least getting rid of it, but it may not eat the arsenic. So the arsenic might remain. I don't know.
Right. That's a good question. I've heard about a bunch of different things, but I don't know what muck eater is. Maybe Chris knows something about what that might be.
I can tell you 10 pounds cost me $160 on Amazon, but wow. You don't use that much of it. And it seems to do a good job in sort of limited areas.
So it's just cleaning up some of the organic matter that you
Cleans
see,
up the
so it's
organic, yeah.
less mucky. So one, that definitely wouldn't do anything for arsenic, as far as it doesn't get rid of arsenic, right? So those muck eaters, I'm guessing it somehow jacks up the microbes so that they consume more of the organic carbon and turn it into CO2. And so that kind of removes that carbon from your system. But the arsenic, being a metal, doesn't volatilize, it doesn't, so it might change form. It's possible that it would definitely have some impact on how low oxygen happens in the lake, because you're getting rid of the organic matter that causes some of that low oxygen. But I don't know enough about that as to know how much of an effect it would have kind of at the lake scale. Because again, if you're cleaning up some of the stuff near your area, it tends to be that it's gonna get transported from other areas as well. And whatever the organic carbon that got eaten up, it's possible that the arsenic's just in the rest of the sediments left behind. But it's a good question. I don't know. I'll have to look into what muck eater is. That's a new one. Leah?
Yeah, thanks, Jim. Thanks for your update. That was very illuminating. And I'm wondering, obviously, I totally understand you guys are kind of at the beginning of looking at the ramifications of this. I know incrementalism from working for local government, but I'm wondering, do you have a sense of how quickly the bioaccumulation is occurring in the tissues of the aquatic organisms? Like, is it a linear kind of, accumulation of arsenic in their tissues, or is it exponential? Kind of like, what time frame are we looking at? I'm guessing, or more of my question is, how sensitive, that chart you had color coded, is that something where those could kind of turn year on year, or is that like a 10-year, kind of thing where we'll look at seeing a difference?
Gotcha. I'm going to answer what little parts I can of that. Some of this we don't know. Arsenic can get expelled from an organism, even and from humans. So it turns out that if somebody, this harkens back to the days of when people used to poison each other with arsenic. But it turns out you actually can get rid of arsenic out of your system, right? So you actually do excrete some of that. Same thing goes with the organisms. So if you were to decrease arsenic that's available to the organisms, the concentration in the organisms should go down relatively quickly. I don't know exactly what that scale is. But if you say similar to like phosphorus, if I go and clear all the arsenic out of the water column, but then the arsenic is still coming out of the sediments, then you're going to just have maybe a short-term effect by getting it out of the water, but then you have this source that's still potentially feeding things up in the top. So that's where it depends on the mechanism of how you're kind of dealing with it as to how long your effects would last. But as far as the organisms themselves shouldn't hold on to arsenic for very long should you get rid of the arsenic that's in their surroundings. At least that's my take on it. As far as they also, because of the fact that the organisms excrete arsenic, they tend to take things up and excrete it at some rate. And so that kind of determines how much they actually hold on to. So it depends on the organism, how they do that. So that's where different fish might do things differently. Even in different lakes, it's possible that if the fish have been living in Lake Killarney for long enough, or especially the lower organisms, they can kind of develop some ability to adapt to the conditions. So it's somewhat hard to predict from lake to lake, but we're trying to learn more about that.
No, that's awesome. Thank you.
Other questions from anybody? Right. Well, did anybody from any of the agencies or people here want to chime in with anything? I'm not trying to put anybody on the spot, but if you had anything you wanted to add before we sign off.
So Jim, this is Marion.
Hi, Marion.
There was the earlier question about risks for swimming in the lakes that we didn't really
Oh, yeah.
touch on too much. And I was thinking about it, and I don't know that I can fully answer it, but you know, what we do know about arsenic is that it's not well absorbed through the skin. The bigger concern is ingesting it. And so a concern for swimmers might be accidentally swallowing the water, or you know, if you get sediments on your hand playing on the beaches or whatever, and it's contaminated sediments, you'd be concerned about accidentally ingesting the sediments. So we would encourage what we call healthy actions. Just, you know, try and avoid swallowing the water, and then to wash your hands off, wash your body off, to make sure that it's not on you so that you don't accidentally ingest it. And those are similar to the same healthy actions that we put out about the arsenic in the soil from the Tacoma Smelter Plume Project, which is what I work on. So thought I'd add that.
Great, thank you. I do know in other lakes that I've worked on, there has been people that take lake water to irrigate their lawns. I'm not sure if it's legal or not, but I've actually seen pump lines run out into a lake before. I've never seen it in Killarney or Steele, but I've seen people run pump lines out and irrigate their lawns with it. That would also, kind of building off of what Marion just said, that would be a kind of, that would increase the potential exposure, because now you're putting that arsenic on your soil where people are playing around and walking into the house and so on. So if you know anything along those lines, it's a good idea to try to let people know that that's not a good practice for lots of different reasons. But again, that's just, I'm not sure if that happens out here, but I have seen it in other lakes that I've worked on. So I know it does happen occasionally.
Hey, Jim, I had a question.
Yes, please, go ahead.
This is Dave Heimer. Do you see any differences, not part of your study probably, but do you see differences in, let's say, the biota of in some of these lakes that have had arsenic for years and years and years, where you have, you know, many generations of phytoplankton reproducing every year? Is there differences in the assemblage or the type of phytoplankton in there? Because it's kind of the so what question. It's like, okay, there's arsenic in the lake, it's cycling, you got different fish. Is it different than anything else? Yes, it's bad for people if you eat at a higher risk of cancer, but has it changed the biota in any way that you can measure?
Right. That's a really good question. We don't know. I would say we just took some initial data just to look at the differences between phytoplankton populations and zooplankton populations in the 3 shallow lakes, so in Bonnie, Steele, and Killarney, that have different concentrations of arsenic in them. The issue with phytoplankton is that the populations change dramatically from week to week or from over the season. And so it's always hard to say with just a few grab samples whether you're seeing a real difference. We do see a difference in phytoplankton populations, but again, we haven't done enough work to know, enough ID work to know whether we're seeing a real difference between those or not. And we don't know enough about the susceptible, without those IDs, there's a little bit of information in the literature that says that some phytoplankton are susceptible to metals and others aren't. And so we might be able to predict something along those lines. But at this point, we just don't have enough information to say conclusively. But that is part of that interest in knowing more about ecological health. If we are changing the population of phytoplankton that are in the lake, how does that affect everything else that depends on the phytoplankton as a food source? Does that start to change the type of zooplankton that grow in the lake, and therefore does that change what fish do well in the lake? So that's all part of that ecological question that we don't know at this point. Yeah, Norm.
Are we likely to see you out here doing any additional research, or are you pretty much done with us, or is your next phase to evaluate the potential to remove some of this gunk or seal it up or whatever you choose to do? Where are you at right now?
Gotcha. But we don't plan to abandon you anytime, Norm. We have too much fun going out on your guys on both lakes. And as you know, we know working on both Steel Lake and Lake Killarney has taught us a lot about what's going on in these shallow lake systems. And so our hope is to actually continue to do some work. And as you saw, some of those things we're hoping to do, we hope to get the support of both Steel Lake and Lake Killarney to continue to do some work out on your two lakes. So I'll email you guys about that. If I know last time you wrote us a letter of support, which was really important for us to get the grant in the first place. And so we're hoping that that would be the same. But we want to see if we can find a way to actually address the arsenic that we found. So that's part of what we're doing in the next probably 2 years from now. This summer, you won't see us much. When COVID, anything out on the lake somewhat interesting. So we'll see a little bit less of us there, but we'll be out doing some follow-up work. We talked with Fish and Wildlife actually about doing potentially some follow-up sampling this summer. So we might see, I'm not sure if we'll be out there or they'll be out there potentially collecting some fish samples to look at. Right now, we're kind of finishing up what we have and then hopefully kicking in the next stage probably 2 summers from now is what the hope is in 2022. But again, if we get to meet in person, we're happy to come out and talk to Lake Killarney folks again in a bigger group. I know this isn't as desirable, but that's what we're hoping to do, is actually to do some more work that doesn't just do the same thing, but actually starts to move forward with what do we do next? How do we deal with this? John, I saw you had yourself unmuted. Did you have something, question, anything else? Okay, great, thank you. Any other questions from anybody in the, well, not in the room, on the Brady Bunch face page here?
Jim, you might be doing this anyway, but I was just going to suggest you let folks know about your publication and when they might see information coming out publicly, when they might get a fact sheet from us, that kind of thing.
Excellent. Yeah, sorry, I thank you for reminding me, BJ. So the work that we did that we just presented to you, we publish those so that we actually get feedback from the people who review those papers. Otherwise, it's our word without anybody checking us on it. So the paper that we wrote about this is in review right now. It's already gone through 1 round of review, and will go through its our final round of review actually by next week. So that means that the publication could potentially come out in the journal that we've sent it to within a couple, it depends on their speed, but a couple weeks or so after that. I would let you know that usually what happens is that gets picked up by some people who monitor anything that's relevant to this region. As you can tell, you see research articles in the newspaper on a regular basis. So you may hear something from a reporter or something asking questions if they can contact you. That might happen, just to let you know. And if anything that you want to do, you can always send them my way if there's something that you don't feel comfortable with, or if they come up with something that you want to know, again, email me and let me know. But just to let you know that that's coming out, and it tends to be that some portion of the press tends to always find those things when they come out, just so you know. Okay. And then BJ, what about fact sheets? Sorry, what were you thinking about there?
We can certainly send kind of a science fact sheet that Lisa has already developed. Right. We should also maybe talk internally. We might be able to put together one that addresses some of the questions that were asked here today as well, kind of an FAQ section. So, but I assume Jim has the contact needed to get that to you all. So yeah, just stay tuned. We'll make sure to keep you as informed as we can as we go forward.
Excellent. So if it's okay, I'll make sure that I get stay in touch with Leah and with Norm, if that's the best way to make sure. And I'll of course include folks from King County, so Chris Knutson and folks, because I do know there's been some interest in what we're doing at Lake Geneva, which is right across the street from Lake Killarney, and other folks. So we just want to make sure that everybody knows what's going on. So we'll try to get that information out to you in a digestible form that's a lot easier to deal with than a scientific paper usually. So hopefully that will help out. But again, if there's any other information that you want to help to explain to other people in your lake resident groups, just let us know and we'll try to work towards that. Happy to do that. Any other questions, comments from anybody?
I just have a question for members of the Lake Association. I'm just curious whether you ever see anybody harvesting snails from the lakes for 8 to 8. I'm seeing heads shaking.
John saying no.
Never seen that. You see crayfish being harvested, but never snails. I haven't.
This particular type of snail was actually imported as a food source, to be grown as a food source. So it's a question we're really interested in.
I've asked that question to numerous volunteers and residents on our lake stewardship about the Chinese mystery snails, and I have yet to hear anybody who knows of anybody that has ever harvested them for food or eaten them. And everybody's like, really? They eat them? And the advice is don't, because they might have a liver fluke that you can get, but we're just wondering.
Yeah, we've seen them chase down frogs, come in with their big lights at night and find frogs and harvest them, but no snails that I'm aware of.
Huh. Well, that's an interesting point, Norm. We like to think that we're trying to think of all the organisms that somebody could eat, and frogs missed me, even though I grew up in Kentucky and I actually eat frog legs quite regularly. So now we're going to have to actually sample some frogs and see what's going on there. Yeah, I thought we had it all covered. But
got turtles.
It's interesting because we've seen the turtles in your lake. They're always kind of a friendly thing to see.
Yeah.
I don't know. Well, it's possible that they would be getting some uptake, but I don't think turtle harvest is. I don't know if that's even allowed or not.
I hope not.
Yeah. My guess is they'd pick a different place and bigger turtles, but I don't know for sure. All right. Well, I don't want to hold you guys any longer. It's actually the only sunny day we're going to have for the next 3 months, I believe. So hopefully you can get outside and enjoy it a little bit. I want to thank everybody for coming. I will send out the info to Leah and Norm, and if anybody else is interested directly, just send me your email contact, and we'll get you the site where this is going to be recorded so that you can share that with other people and any other info that as we go forward. I want to thank all of the folks from the different agencies in the City of Federal Way that came today. I really appreciate it. And if anybody has any additional questions, again, anybody should feel free to contact me, and I will pass you on to the people that actually know what they're talking about. So we'll go from there. Thank you all very much. Have a great day.
Thank you.
Talk to you later.
Thank you. Appreciate it. Thank you. Thanks, Leah.